Although global life expectancy has increased, the timing for reproductive aging in women remains unchanged. And that's why in IBA we are really focused and committed to studying the reproductive aging phenomena. Because we know that most of this age effect is explained by an increase in prevalence of embryonic aneuploidy with maternal age. And we also know that there is a relation between shortening of telomeres in polar bodies and the presence of aneuploid embryos. Therefore, telomere measurement seems to be a potential predictor of ovarian function and of oocyte and embryo quality and of clinical outcomes as well. However, in clinical practice, it is rather impractical to measure telomeres in the ovaries. So we will need an accurate surrogate for telomere length in the follicular cells. Thus, we designed this proof of concept study to assess if telomere lengths in peripheral blood could be a reliable surrogate for telomere length in follicular cells, and therefore an indicator of the mature oocyte's reproductive competence. So we selected a homogeneous population of young healthy egg donors. On the date of the pickup, we isolated Cumulus and granulosa cells, and then we isolated genomic DNA for telomere length measurement in the three cell types: leukocytes, Cumulus cells, and granulosa cells. But we didn't observe a positive correlation between telomere length in the three cell types, suggesting that telomere length in leukocytes is not an indicator of telomere length in the follicular cells, according to our results. But interestingly, when analyzing the average telomere length measurements, we did observe that telomeres in Cumulus cells were significantly longer than telomeres in leukocytes. We believe this is an interesting finding. Maybe this data argues for the existence of a mechanism of coping against telomere attrition in the ovary. So we are really encouraged with this research line. We will continue studying telomere biology in order to understand the telomere implication in the ovarian physiology and reproductive outcomes.